Chapter 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cell, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. 10.1 Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also take place. All these all these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cell content containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesis the other constituent of the cell, and eventually divides into two daughter cells is termed cell cycle. Although cell growth is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. 10.11 Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by a human cell in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases. The first, that is interphase, second, M phase. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phase. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts for more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, that means karyokinesis, and usually ends with the division of cytoplasm, that means cytokinesis. The interphase through called the resting phase is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, S phase and G2 phase. G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S, or synthesis phase, marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell double. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosome at G1, even after S phase the number of chromosome remains the same, that is 2n. In animal cell during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the animals do not appear to exhibit division, that is herd cell, and may other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive stage called quiescent stage of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to these where haploid cells divide by mitosis. For example, male honeybee. Against this, the plant can show mitotic division in both haploid and diploid cell. From your recollection of example of alternation of generation in plant in chapter 3, identify plant species and stages at which mitosis is seen in haploid cell. 10.2 M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cell is the same, it is also called as equitational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division. That means karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, and telophase. 10.21 Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecule formed are not distinct but intertwined. Prophase is marked Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation, that is given in figure 10.2a. The centrosome, which had undergone duplication during S phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristics event. First, chromosomal material condensed to form compact mitotic chromosome. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Second point, centrosome, which had undergone duplication during interphase, begins to move towards opposite pole of the cell. Each centrosome radiates out microtubules called esters. The 2 esters, together with spindle fiber, form mitotic apparatus. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. 10.22 Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell by this stage. Condensation of chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. Small disc-shaped structure at the surface of the centromere are called kinetochore. This structure serve as the site of attachment of spindle fiber to the chromosome that are moved into the position at the center of the cell. Hence, the metaphase is characterized by all the chromosome coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as the metaphase metaphase plate. The key feature of metaphase is first, spindle fibers attached to kinetochore of chromosome. Second, chromosome are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. 10.23 Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosome of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of the chromosome trailing behind. Thus, anaphase is characterized by the following key events. First, centromeres split and chromatids separate. Second, chromatids move to opposite pole. 10.24 Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows the following key event. First, chromosome cluster at opposite spindle poles, and their identity is lost as discrete element. Second, nuclear envelope develop around the chromosome cluster at each pole, forming two daughter nuclei. Third, nucleolus, Golgi complex, and ER reform. 10.25 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In an animal cells, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation start in the center of the cell and grow outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of simple precursor called the cell plate that represent the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium, that is liquid endosperm in coconut. 10.3 Significance of mitosis. Mitosis, or the equitational division, is usually restricted to the diploid cells only. However, in some lower plants and in some social insect, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insect? Mitosis usually result in the production of diploid daughter cells with individual genetic complement. The growth of multicellular organism is due to mitosis. Cell growth result in disturbing the ratio between the nucleus and the cytoplasm. If therefore becomes essential for the cell to divide to restore the nucleus-cytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic division in the meristematic tissue, the apical and the lateral cambium result in a continuous growth of plant throughout their life. The production of offspring by 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. These specialized kind of cell division that reduces the chromosome number by half result in the production of haploid daughter cells. This kind of division is called the meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organism, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plant and animals. These lead to the formation of haploid gamete. The key feature of meiosis are as follows. Point number 1. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Point number 2. Meiosis first is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Point number 3. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatids of homologous chromosome. Fourth point, 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following phases. 10.41 Meiosis first. Prophase first. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following five phases based on chromosomal behavior, that is lipotine, zygotin, pachytine, diplotin, and dikinesis. During lipotine stage, the chromosome become gradually visible throughout the lipotine. This is followed by the second stage of prophase. Prophase first, called zygotin. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosome. Electron micrograph of this stage indicate the chromosome synapsis is accompanied by the formation of complex structure called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is called bivalent or a tetrad. However, they are more clearly visible at the next stage. The first two stages of the prophase are relatively short-lived compared to the next stage, that is pachytine. During this stage, the four chromatids of each bivalent chromosome becomes distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occur between non-sister chromatids of the homologous chromosome. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytine, leaving the chromosome linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of crossover. These X-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinesis, the nucleolus disappear and the nuclear envelope also break down. Dikinesis represent the transition to metaphase. Metaphase 1. The bivalent chromosome align on the equatorial plate. The microtubule from the opposite pole of the spindle attached to the kinetochore of the homologous chromosome. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromere. Telophase 1. The nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called as dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between two meiotic divisions is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase second, a much simpler prophase than prophase 1. 10.42 Meiosis second. Prophase 2 Meiosis second is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to Meiosis first and Meiosis second resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosome again become compact. Metaphase 2. At this stage, the chromosome align at the equator and the microtubule from the opposite pole of the spindle get attached to the kinetochore of sister chromatids. Anaphase second. It begins with the simultaneous splitting of the centromere of each chromosome, allowing them to move to an opposite pole of the cell by shortening of microtubules attached to kinetochore. Telophase second. Meiosis ends with the telophase second, which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organisms, even though the process per se paradoxically result in reduction of chromosome number by half. It also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution. According to the cell theory, cell arises from pre-existing cells. The process by which these occur is called cell division. Any sexually reproducing organism starts its life cycle from a single cell zygote. Cell division does not stop with the formation of the mature organism but continues throughout its life cycle. The stages through which a cell passes from one division to the next is called the cell cycle. Cell cycle is divided into two phases called interphase, a period of preparation of cell division, mitosis, and phase, the actual period of cell division. Interphase is further divided into G1 as G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. As phase mark the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase, telophase. Chromosome condensation occurs during prophase simultaneously. The centrioles move to the opposite poles, the nuclear envelope and the nucleolus disappear, and the spindle fibers start appearing. Metaphase is marked by the alignment of chromosome at the equatorial plate. During anaphase, the centromere divide and the chromatids start moving towards the two opposite poles. Once the chromatids reach the two poles, the chromosomal elongation starts. Nucleolus and the nuclear membrane reappear. This stage is called the telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis thus is the equatorial division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in the diploid cells which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by half while making the gamete. In sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value in the parent. Meiosis is divided into two phases, meiosis first and meiosis two. In the first meiotic phase division, the homologous chromosome pair to form bivalent and undergo crossing over. Meiosis first has a long prophase which is divided further into five phases. These are lipotine, zygote, packitine, diplotin, dikinesis. During the metaphase, the bivalent arrange on the equatorial plate. This is followed by the anaphase first, in which the homologous chromosome move towards the opposite poles with both their chromatids. Each pole receive half the chromosome number of the parent cell. In telophase 1, the nuclear membrane and the nucleolus reappear. Meiosis second is similar to mitosis. During anaphase second, the sister chromatids separate. Thus, at the end of meiosis, four haploid cells are formed. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement. So मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय, एंड जस्ट स्टे ट्यून्ड विद बायोलॉजी क्रिटिक एंड एंजॉय द नॉलेज. बाय.